Hello ladies and gentlemen, and welcome to Lecture 7, Respiration and Metabolism. So in our last lecture in photosynthesis, we made carbs, we made glucose. Respiration is the opposite. Respiration is taking complex molecules like glucose and turning them into energy. So you take glucose and oxygen, which you breathe in, and they turn it into carbon dioxide, water, and a bunch of ATP. This doesn't require energy to be put into the system because it increases the entropy. Now everything undergoes respiration, even plants. Plants make their sugars with photosynthesis, but they break down some of them in order to keep the plant going. So, overview of respiration: glucose, ADP, and oxygen turns into carbon dioxide, ATP, and water. Here is a cartoon showing you an overview. We're going to talk about 4 major steps of respiration. First is glycolysis. Then in here we have acetyl-CoA formation, the Krebs cycle, and electron transfer phosphorylation. First is glycolysis. This is a series of reactions that begin carbohydrate breakdown. This happens in the cytoplasm, and it starts when the glucose enters the cell through a transport protein. An enzyme then immediately phosphorylates it. Wonder why that is. Well, remember with diffusion we talked about how different things follow their own concentration gradient? Now we've changed glucose into glucose phosphate. So phosphate is going to have a different concentration gradient than glucose. So if there's less glucose outside of the cell than inside of the cell, and we didn't phosphorylate it, the glucose could blip right back out again. The phosphorus keeps it inside the cell. Another ATP is burned or spent to add another phosphate, and the glucose spontaneously breaks in half into 2 3-carbon sugars we call pyruvate. This makes 2 NADH, remember hydrogens, we can use those later, and we gain 2 more ATP. These ATP are regenerated when the phosphates fall back off pyruvate and go back on ADP. Then substrate level phosphorylation occurs, and 2 phosphate groups are directly transferred from a substrate to ADP, making ATP. So what I want you to get out of glycolysis is happens in the cytoplasm. It's the start of respiration. In the end, you get 2 ATP and 2 NADH. If you're planning on doing the extra credit for this unit, I highly recommend printing it out and following along. Okay, so here's a cartoon showing you what just happened. Here's the glucose, add a phosphate, add another phosphate, that burns 2 ATP. Then it falls in half, you get your NADH. Excuse me. You get 2 ATP produced and then another 2. So you make up for the 2 you burned up above, and you end up with a net gain of 2 ATP and 2 NADH. And you have 2 molecules of pyruvate ready to go to the next stage. Now we're going to go into the mitochondrion. The mitochondrion is a lot like the chloroplast in that it has a bunch of membranes folded in on itself. These membranes are excellent places to do chemical reactions like respiration. The next step is where the acetyl-CoA happens, or the acetyl-CoA gets formed. So the pyruvate gets taken into the mitochondrion. We make the acetyl-CoA and do the Krebs cycle. So how do you make the acetyl-CoA? The 2 pyruvate enter into the inner compartment of a mitochondrion. And an enzyme splits them into a 2-carbon acetyl and 1 molecule of carbon dioxide. So we're doing this twice because we have 2-pyruvate from the last step. So we've given off 2 carbon dioxide. We've broken our 6-carbon sugar into 2 2-carbon acetyls. So the acetyl then combines with coenzyme A and becomes acetyl-CoA. You get hydrogen ions released and 1 molecule of NADH is formed. So the key part about making the acetyl-CoA is you lose 2 carbons as carbon dioxide and you make 2 molecules of NADH, 1 for each pyruvate. Now we've got our acetyl-CoA and we're ready, so this was the step here, and we're ready to enter into the Krebs cycle. The Krebs cycle is another pathway with a bunch of enzymes in it, and it breaks acetyl-CoA down into carbon dioxide. The last reaction regenerates the substrate of the first, so it is a cycle. And the first step in the cycle is the 2-carbon acetyl-CoA binds to a 4-carbon sugar to make citric acid. That's why we call this cycle either the Krebs cycle, after the gentleman who discovered it, or the citric acid cycle, after what's made. At the end of the cycle, remember we're doing the cycle twice because we started with 2 acetyl-CoA, we wind up with 6 carbon atoms lost as carbon dioxide, 2 ATP formed, 6 NADH, and 2 FADH2. The coenzymes that are reduced in the Krebs cycle carry the energy that drive the endergonic reactions. So here's the schematic of that again. We've got an NADH here. Here we get the FADH2, ATP, some more NADH. This brings us to step 4, and this is the money step. So now we don't have any sugar anymore. We don't have any carbons. We just have NADHs and FADH2s. This step involves an electron transport chain in the membrane of the mitochondria, just like the one in chloroplasts for photosynthesis. So no sugar or carbons here, just enzymes bound to the mitochondrial membrane and redox reactions. So the coenzymes FADH2 and NADH donate their electrons and hydrogen atoms to the electron transfer chain. That's what they're doing. Think of the NADH and the FADH2, the quarter to start this game. So you need that in order to do electron transfer phosphorylation. Just like in photosynthesis, electrons are transferred from one enzyme to another in a series of redox reactions. And this transfer drives the pumping of hydrogen ions into the outer mitochondrial compartment. Once again, just like in photosynthesis, the hydrogen ions can only leave the outer mitochondrial compartment by traveling through an ATP synthase. This enzyme adds a phosphate to ADP, and at the last stage of the chain, oxygen accepts the electrons and combines with the hydrogen ions, forming water. So it's the opposite of photosynthesis. In photosynthesis, you split up water to get oxygen. In respiration, the oxygen accepts an electron and combines with the hydrogen to make water. In the end, you get 32 molecules of ATP. That's why this is the money step. That is a lot of energy. So here's a schematic of this happening. It looks a lot like the one from photosynthesis. The NADH and the FADH2 donate their electrons, go through the electron transfer chain, and it pumps hydrogen ions into this little compartment. The only way the hydrogen ions can leave is through the ATP synthase. So they go out there, and when each one leaves, a molecule of ATP is made. And this electron here has to go somewhere. It winds up combining with oxygen, and the oxygen combines with hydrogen, and you get water as a byproduct. So back to the overview again. If you can talk your way through this step through this diagram, you know enough to take my exam. I'm not worried that you know the names of all the little intermediates in the cycle. I want you to know what happens in glycolysis, what the purpose of the Krebs cycle is, and where you get most of the ATP. Most of the ATP comes from electron transfer phosphorylation, and you can only run that with the NADH and the FADH2 that you made in the Krebs cycle. So the pathway we just described requires oxygen to work. Therefore, it's known as aerobic respiration. But what if there is no oxygen? What do we do then? Well, if there's no oxygen, you can still do glycolysis. Glycolysis can happen regardless. And then, if you don't have any oxygen, you can do anaerobic respiration. This is also called fermentation, and it doesn't generate nearly as much ATP as aerobic respiration. In alcoholic fermentation, this is an anaerobic form of respiration. You do glycolysis, and you don't go into a Krebs cycle or the electron transfer chain. You generate some NADH so that glycolysis can continue, and you end up with ethanol. So you break up the glucose, you get the pyruvate at the end here. The pyruvate gets broken down, and you burn, you burn this NADH in order to make this a little cycle here. You burn the NADH, and the byproduct is ethanol. Ethanol is also called ethyl alcohol. It is the part of beer and wine that gets you drunk. In beer and wine making, it's yeast that does alcoholic fermentation. And that's why, if you've ever tried to brew, you know that you're not supposed to let oxygen in after a certain step, because if there's oxygen, then the yeast will switch to aerobic fermentation and you won't have beer. Now, there's another pathway that can happen after glycolysis as well. This is another kind of anaerobic respiration called lactate fermentation. The end product of this is lactate, which is a slightly different sugar. This is what the bacterium Lactobacillus acidophilus, there we go. That is the bacteria that turns milk into yogurt and cheese. So it does anaerobic, that bacterium does anaerobic respiration, and it makes yogurt and cheese. This also happens in your own body inside certain muscle cells. This is why if you're trying to run really fast and you're kind of out of shape, you'll get that stitch in your side. That's the burn of lactate acid in your muscles. So in the human body, and in the fish body or the bird body for that matter, we have two main kinds of muscles, red muscles and white muscles. Red muscles, which are these here, are also called slow twitch muscles. They perform aerobic respiration. They have lots of myoglobin, which holds oxygen. Myoglobin is a protein that stores oxygen. So that helps it with the aerobic respiration. The white muscles, on the other hand, are fast twitch. They do anaerobic respiration, so they do short bursts of activity. So if you're sprinting, you're using your white muscles. If you're doing a long controlled run, you do your red muscles. So before I go away, so think about it this way. You've got your domestic chicken. Your chicken doesn't do, it does some running away from foxes and dogs and that sort of thing, but it doesn't really fly. So chicken breasts are white muscle or white meat. Now think of a duck. Ducks migrate, or at least they're supposed to. So they fly hundreds of miles. They have a lot of red muscles or slow twitch muscles. That's why if you eat breast meat on a duck, it's dark meat. I found that out the hard way. Yeah, duck l'orange, delicious, no white meat. So, what does this mean for you after you eat? How does your body take the glucose and do respiration? Well, as you digest your food, aerobic respiration kicks into high gear. If you eat too much sugar that your body needs at that instant, it gets made into glycogen and stored in your liver. The glycogen reserves are typically used in 12 hours, so that's what keeps your blood sugar up even when you're not eating. Now, if you eat too much sugar, that you've used up all that you need, your glycogen reserves are full, then acetyl-CoA can actually be diverted from the pathway that makes from respiration to the pathway that makes fatty acids. So the acetyl-CoA gets made into fatty acids, and the sugars you eat get turned into fat. This is why, even if you ate an entirely fat-free diet, you could still gain weight. I believe that's on your essay number 3. So during meals, excess glucose is converted to glycogen or fat, and between meals, the glycogen gets degraded, the glucose subunits enter glycolysis, then the fats are degraded to fatty acids once you're out of sugar. And then once you've burned all the fats, you start to burn the proteins. And all of this can go back into the Krebs cycle or acetyl-CoA, so this can all go into the pathway we talked about. So here's a little chart showing you where the food you eat goes and how it enters the pathway that we talked about, respiration, with glycolysis in the Krebs cycle and electron transfer phosphorylation. So when you eat fat, some of its fatty acids get made into acetyl-CoA, go into the Krebs cycle. Some glycerol gets made into PGAL, goes into glycolysis. All carbohydrates, boom, straight to glycolysis. Proteins can get made into pyruvate, they can get made into acetyl-CoA, and they can get made into intermediates in the Krebs cycle. So no matter what you eat, even if you're not just eating glucose, all of your food gets taken into this process. Thank you, and good luck studying for exam number 2.